Hi everyone, my name is Parisa. I'm a PhD candidate in Biomolecular Sciences program at Laurentian University. I'm performing my PhD thesis under the supervision of Dr. Alexander Moyes at Nazem University. Here I am pleased to talk about some of the results of my PhD project. And the title that I want to talk about is Retinoic acid regulates commitment of osteoprogenitor cells during bone development. As a disclosure, there is no conflict of interest, and this program has not received financial or in-kind support. As you may know, retinoic acid is one of the main bioactive metabolites of vitamin A that display its role by binding to a group of nuclear receptors known as retinoic acid receptors and regulate expression levels of more than 500 genes. Retinoic acid is a critical morphogen that in collaboration with other morphogens define the patterning of the body during embryonic development in a time and dose-dependent manner. Therefore, any change in the concentration of retinoic acid is associated with the pathogenesis of a wide range of birth defects. Concentration of retinoic acid regulates by a complex network of enzymes, transcription factors, and binding proteins. That my project focused on investigating the role of an important enzyme, a short-chain dehydrogenase enzyme known as NHRC. This enzyme plays a critical role in preventing production of excess amount of retinoic acid. Now, the reason for choosing this enzyme is that our clinical and in vivo studies demonstrated that change in the activity of this enzyme is embryonic lethal or causes a broad range of birth defects, including craniosynostosis. Therefore, in this study, we aim to investigate how change in the activity of this enzyme affects the process of osteogenesis. I employed a pre-osteoblast cell line and make different cell models of retinoic acid deficient and excess models. Some of these techniques are CRISPR-Cas9 and also using different chemicals such as Win18446, which inhibit an enzyme that has a role in production of retinoic acid. Here are provided you with a summary of results I got in this study. As you see, I investigated mineralization and differentiation of pre-osteoblast cells in different states of osteogenesis in retinoic acid deficient and excess models. As the results demonstrated here, by knocking out DHR3 and making an endogenous retinoic acid model, the late-stage osteogenic markers upregulated significantly in the early states and deposited calcium earlier than expected in the osteogenesis process. This result showed us that excess amount of retinoic acid caused early differentiation, which is in agreement with clinical data, and early fusion of suture, which is one of the main reasons for the pathogenesis of craniosynostosis. However, results for retinoic acid deficient model are completely in opposite and cause delayed differentiation and mineralization. In summary, results of this study highlighted one more time the critical role of DHRS3 in the regulation of retinoic acid signaling during embryonic development. And in this study, we specifically work on osteogenesis. I would be more than happy to take any question and have more discussion about the result of this study. Please feel free to reach out. Thank you.